All right. Hey, everyone. My name is Jared Cole, and I'm a PhD candidate at UT Austin, working with Mark Kirkpatrick and Arbel Harpak. And today I want to present some results of my project where we investigate signals and consequences of sexually antagonistic selection in the human genome. Now, as we know, selection favors different phenotypes in females and males. And this makes sense because the sexes often occupy very different reproductive roles, which require different phenotypes. So as you can see with um, these deer on the left-hand side here, with the male having very large antlers, and this stag beetle on the right, where males but not females have these massive mandibles. More than that, though, selection can act on the same regions of the genome within both sexes and can act on alleles that might be good for one sex but bad in the other. And that's what's known as sexually antagonistic selection, which I'm going to abbreviate as SAS. So, why do we care about SAS? Well, it drives the evolution of sexual dimorphism. It has large demographic consequences, it can maintain genetic variation for diseases, and we think it's important in driving the origin and evolution of sex chromosomes. So SAS isn't limited to non-human species, of course, and we can see that males and females are different in our own species. And I should say by male and female in humans, I'm referring to individuals that are XX or XY karyotype, and that, in addition to that, also identify as male and female, just so we're clear on that. But we think that there's ongoing selection in humans for different phenotypic optima in the sexes. So this is a study from 2010, for example, that shows the distribution of heights in the UK for females here in the open histogram and for males here in the blue. And this solid curve over here represents the number of children for females. And the dashed curve is the number of kids for males. And that shows pretty clearly that females that are on average 3 centimeters shorter leave 15% more kids than the average female. Whereas for males, if they're taller than the average, they leave more offspring. So we think SAS is definitely occurring in humans and is ongoing. There are, however, some major difficulties for studying sexually antagonistic selection at the genomic level. Most population genetic tests for selection pick up on signals that accumulate over many generations. So if we use a test, for instance, based on the allele frequency spectra, like Tajima's D here in blue, to look for a selective sweep, it's going to take many generations of selection before a detectable signal emerges. Unfortunately for SAS, Mendelian rules of inheritance mean that any signal of divergence between the sexes gets erased as males and females are shuffling their autosomal genomes each generation. So what do we do about that? What we have to do is study selection acting in the current generation, or what we call selection in real time. So the basic idea is that at conception, the sexes are equal in allele frequency on the autosomes. And this PF minus PM is the difference in their allele frequencies, which is at 0. But once sexually antagonistic viability selection has acted in each sex, there are now allele frequency differences between the sexes on the autosomes in the adults and at adulthood. And many studies have, both at our lab and many others, have used this approach to investigate the presence of SAS in humans and other species and have found positive signals utilizing allele frequency differences between the sexes. But despite the evidence that has accumulated for SAS in humans, there has been some controversy in the literature with heavy skepticism that SAS is that it's ongoing in the genome. That the gist being that most of the signals that we're picking up of SAS are in actuality bioinformatic artifacts. So we still have many outstanding questions about SAS, such as how strong and widespread is selection across the genome? How is it related to phenotypic traits? Do we see life history trade-offs between survival and fecundity? And what are the demographic costs to this kind of selection in the population in terms of mortality? So now it's our turn to tackle these questions. And one way to really increase our ability to detect subtle signals of selection is to use a very large data set. So the largest genomic data set currently available is the UK Biobank, which has over 500,000 participants in the UK. And we focus on a subset of over 500,000 phased SNPs for over 250,000 individuals, which is important to the method that we're using to estimate signals of SAS. Now, the reason we want phased haplotypes is because we employ a method that will improve our estimation of SAS at each SNP. So here's the basic idea. If we have a chunk of chromosome here and a target site under SAS with the neutral SNPs nearby in blue, what happens is this. So what I'm showing you here is that this vertical axis, this is the sex difference in allele frequencies, which at conception before SAS acts is 0, because the sexes are equal in allele frequencies at conception, as we've seen. But once viability selection acts on the target site, there will be sex differences in allele frequencies at that target site, but also at the sites nearby as well, because they are going to hitchhike along with the target of selection. So we can leverage information not only at the target SNPs, but at the SNPs nearby using phased haplotype data. So toward that end, we developed a proper likelihood model that will estimate the strength of SAS acting on individual sites. And this approach is different from a descriptive statistic like FST that will estimate divergence in allele frequencies between the sexes, but instead it'll fit a model, right, that gives us an estimate of how strong SAS is acting at a site by way of a selection coefficient. So when we do that, what do we find? Well, let's take this first panel here on the left. This is SAS on viability. We have what I'm showing you here on the horizontal axis is the estimated strength of selection for SAS from 0 to 1%. And these dots here are showing whether there's an excess or a deficit of SNPs showing SAS at a given strength. So these show a statistically significant excess of SNPs at higher estimates relative to a null distribution. And that null distribution is generated by essentially randomizing the sex labels. And conversely, there is a significant deficit of SNPs at lower strength relative to the null for viability. The UK Biobank has data for the recorded number of children for each participant, which is a good proxy for lifetime reproductive success. And that allows us not only to examine allele frequencies in the adults caused by viability selection, but also to estimate SAS on fecundity as well. So for fecundity here in yellow, we see a similar pattern where we can see a statistically significant sort of deficit of the SNPs in the lower bins relative to the null. And if we combine viability and fecundity selection to estimate the total amount of SAS occurring in the genome, we see a very similar pattern, an enrichment at higher selection coefficients and a deficit at lower ones. So all of these results are showing strong evidence for significant genome-wide signals of SAS. Another pattern that we found was a negative relationship for viability and fecundity. So here on the left-handed panel, we calculated z-scores for each of our selection coefficients via bootstrapping. And if we bin our fecundity estimates on the horizontal axis here and plot our viability estimates on the vertical axis, we can see a significant negative correlation here between them. We also see a subtle negative relationship when looking at the selection coefficients themselves. And what this suggests is that there is evidence for a classic sort of life history trade-off between survival and reproduction. Using the UK Biobank, we have access to a lot of phenotypic data and also GWAS summary statistics for this data set. And we use that to examine SAS on 27 traits. And one step that we want to really do is connect SAS acting on the SNPs to SAS acting on the phenotypes. And one way we can do that is by modeling how sex differences in SNP effects are related to the strength of SAS on those SNPs. So what we developed here is a model which relates the selection coefficient, which we're seeing here in this y-axis, to SNP effects on traits in males and females separately. These effects are obtained from a GWAS. And so given this, we perform a weighted sort of regression, like that's shown here on arm fat-free mass, of the estimated value of our selection coefficients against the sex differences in the effect sizes, which is this horizontal axis here. And the slope of that line is what represents the intensity of SAS acting on SNPs that affect the trait, on the alleles that affect that trait. So we use that method. We estimate the intensity of SAS on alleles acting on those traits. And with those regressions, we can calculate a z-score for sexually antagonistic selection, seen here on the horizontal, where the positive z-scores here are indicating selection favoring larger effects in females, the negative scores indicating the opposite. What we see here is a snapshot of the largest signals. The red dashed line here is representing, indicates significance. And we can see that the signals are strongest for many of these body size related traits, but are significant under fecundity for arm fat-free mass. And what we're demonstrating, I think, here is, or what this demonstrates is that SAS does operate on alleles that affect many traits. Okay, so the fact that there are adult allele frequency differences between adult males and adult females means, due to SAS on viability, means that there must be some kind of selective death resulting from selection. So that means that viability is reduced for individuals carrying alleles adapted to the other sex. And so what we want to know is how much mortality is caused by SAS. So we can get at this question by using our estimated selection coefficients and their minor allele frequencies to perform simulations where we can optimize to find best fit parameters for the fraction of SNPs that are under SAS. So we use a method like approximate Bayesian computation, for example. And as we can see here, this is a heat map from an ABC from some early runs where this vertical axis represents the fraction of SNPs estimated to be under SAS F, and the horizontal axis, the selection coefficients, or how strong selection is acting. And this white dot here, is the maximum posterior likelihood, or our best fit parameters. And we can see that close to 1 in 100 SNPs, this F here, is close or are under SAS with an S, or selection coefficient, of about 1%. And so using that information, we can calculate for the first time the mortality burden that's imposed by SAS on viability. And we estimate that we have a reduction in survival to be between 1 in 100,000 to 1 in 10,000 humans due to the action of sexually antagonistic selection on viability. Okay, so that basically, to conclude here, we've demonstrated that SAS occurs, acting on viability and fecundity, occurs in the human genome, that it is highly polygenic with thousands of sites involved. We've discovered that there is a noticeable trade-off between viability and fecundity, that alleles under SAS underlie complex human phenotypes, and that SAS acting on viability causes a measurable mortality load in the population. And with that, I'd like to thank my advisors, Mark Patrick, Barbara Harpak, as well as all my collaborators on this project. So thank you, everybody.